there. I'm Sammy Liao.
Hello, I'm Kurt Von Onreed, and this is Escape of Animals. So Escape of Animals will mainly be talking about GM salmon. It's when biotechnology is applied to salmon and they create genetically modified salmon, which stands for GM salmon. This creates positive results for humans, which means more income. But when these animals escape, it's kind of releasing bioweapons in that these fish can easily take over the native fish and the local ecosystem and it will have negative effects. So we'll explain that more in depth in this video. All right, so to understand, just get a little background on biotechnology. We have to just recap the central dogma briefly. DNA to mRNA to tRNA to protein. DNA codes for the protein, which codes for the gene. So in biotechnology, when you use a substitution mutation, you also substitute a section of a protein by changing the amino acid sequence. And this is actually intentional substitution. So, like in the diagram you see here, you're changing that you're substituting that blue section of DNA into the red DNA. It's kind of like substituting in that whole section, that whole amino acid sequence in the protein below. And this is the diagram of how the salmon are actually bioengineered. You start out with the Chinook salmon protein coding sequence. You take an ocean pounce antifreeze protein. You put that all into a virus, and the virus injects the DNA into the fertilized egg. Keep in mind that the virus isn't rendered benign, so it doesn't harm the fertilized egg. So just to go over the sequence, the commonalities and difference between GM salmon and pea glow is how we'll convey our central message a little bit more, how we'll go a little bit more in depth. One of the commonalities, application of biotechnology, you change the DNA sequence to get a different protein, and you get a different gene. And obviously there are dangerous effects if escape occurs. For salmon, it's the local environment. It will have harmful impacts on the ecosystem by wiping out the native fish. In terms of pea glow, the bacteria, if someone were to get infected, would cause salmonella to spread, and that's certainly something nobody wants. There's also a debate on whether this is ethical or not. We have a political cartoon on this slide here. They actually call these GM salmon Frankenfish, and it's because you're kind of making life, you're engineering life sort of, and it's really all for money sort of. You're making a fish bigger and making it mature quicker just for money, by getting more of a food surplus out of the catch. So really huge debate on whether that's ethical or not. And of course you have to keep these organisms alive in your experiment. This is what we briefly went over earlier, like the procedure. Different genes are used. You have prokaryote, eukaryote, so different organism type, and you have different means of getting the DNA sequence into the organism. So now Sammy will take over here with what we're actually going to go into now, which is the differences in what happens when these organisms actually escape.
So here we go. So basically, when the GM salmon escapes, it causes species not only to go extinct, not only because via competitive advantage, but also because there's something called a Trojan gene that spreads throughout the population, that it interbreeds with like the native populations and spreads this gene, which we'll go into later. And for the pea glow, when it escapes, it's really dangerous because not only does it actually harm humans directly and bacteria infections and gives you like all sorts of nasty stuff, it's ampicillin resistant now too, which means it's antibiotic resistant, and it's very difficult to cure if you like get an infection. So here you can see the many ways GM salmon can escape. You can have like the net simply collapse, and you can have the net gets caught in the propeller, the net breaks, and even the juveniles could escape because even GM salmon's juveniles are ridiculously small, and they could slip through the net and get into the ecosystem that way. So you could see they could escape.
And even if one GM salmon escapes, it could still be able to reproduce as it has high biotic potential. So definitely important to constantly monitor the net and make sure it doesn't have any breaks in it.
Okay, so here now we're going into the actual Trojan genes. So basically the deal is with the GM salmon is that they're like 4 times as large as like a regular salmon, but they have like a 4 times greater chance of being able to reproduce, and they can interbreed with the native salmon population. But the thing is, they also only have like a 2/3% chance, like a 66% chance to survive to maturity, which means that even though you get this weird situation where it's the survival of the least fit, where these genetically modified salmon reproduce, but they don't actually stick around very long and they don't really survive. So the native species of salmon quickly goes extinct pretty much, as you can see in the graph.
Yeah, and once again, that's called the Trojan gene.
So here you can see why we don't want E. coli to escape. Gives you all sorts of nasty symptoms: severe diarrhea, abdominal pain, malaise, and we can't even treat it since we engineered it to be antibiotic resistant. So you can see how they compare to GM salmon is like twice as long and like almost 3 times the weight, and we're exactly alive for the same amount of time. So here you can see the E. coli, and all the way on the right is the E. coli we engineered to glow basically, and it's also ampicillin resistant as well. So in the end, basically as a recap, biotechnology basically can have positive results for humans, but you have to make sure that you maintain them and keep them safe in a lab environment, because if you release them into the wild, it can be very dangerous indeed. We basically release bioweapons.
We hope you enjoyed the video. If you'd like to do some further research, we have a works site here. Thank you very much.